Okay. So we've got like a long muscle fiber, right? And it's divided up into Z discs. Like here's a Z disc, here's a Z disc, and it keeps going. So I'm just gonna kind of draw one sarcomere, one Z disc to one Z disc. And there's stuff going in between. What's something that you know about?
What's running this way?
Yeah, running in between them. Some stuff comes off here, stuff comes off this one, they kind of interact with each other. So anything you can think of.
I'm guessing it's just going to be muscle filaments, so actin and myosin and...
Yeah. Exactly. So there's a protein coming off of each side of the Z disc. And on that protein is exactly what you said, actin. And it's kind of like a double helix. I'm drawing these little spherical balls, but they wrap around kind of.
Got it.
Around.
So it's this diagram thing
here. Yeah, there it is, just like that. And then you said the myosin. There's a protein called titin, which we talked about. Right here, titin. Titin.
Yeah. So titin is just the most renowned protein in a muscle fiber then? Is that the deal there? Or what's the reason for knowing specifically titin?
The myosin heads are attached to it, and they're obviously they are a huge part of muscle contraction.
Because of the cross bridge and stuff.
Yeah.
Like that.
So that has to do with, and this, I forget, this protein right here that these actin sit on has a name. What the heck is it? It's gonna drive me crazy. There's titin and something. Not super important, but it's just gonna bother me. Okay. So myosin heads, what else we got? We have our actin, our myosin heads. There's two more things that sit on this protein strand right here, right there.
So are those myosin heads gonna bind to something up there? Or are they gonna, where are they gonna bind?
These myosin, yeah, they'll eventually bind to the actin.
The active sites, right?
Yes, yeah.
Okay. So the active sites are like covered up, aren't they, or something like that?
Yeah.
That's like getting to the slide and fill them all.
What are they covered by?
Tropomyosin?
Yeah, perfect. So these little sheath coverings here I drew, these little lines, yeah, they are tropomyosin. And there's one more thing on top of these guys. You mentioned it before.
Troponin?
Yeah. And what do they have going on with them? What are they like?
Calcium?
Yeah, I'll blow it up really quick. So we've got our 3 different subunits of the troponin. It's like Mickey Mouse.
Yeah, so it always binds to the tropomyosin and it always...
So one of it likes tropomyosin. Another part likes what?
The actin. And the other one is the calcium. But the calcium isn't always present, right?
Correct.
So just those two are always bound, but the other one isn't.
So yeah, so it's attached to, there's the actin here and the tropomyosin. It's close to those, it's happy, right?
Okay.
So when your muscle is relaxed and there's nothing going on, it's in this state. There's no contraction. Everything is kind of just hanging out, right? But if we want to contract a muscle, we want these Z discs to get closer together, right? Because that's when our muscle shortens.
Yeah.
And that happens by...
So those slide and move, and then because of that, the myosin head's attached to the active sites because the tropomyosin slides. What else slides though? Is it just...
So let's start from the beginning. So what needs to come in order for this to even start? What molecule?
Sodium?
No.
But it's probably calcium.
Calcium, because this guy likes calcium, right?
Okay.
But there's no calcium. So calcium comes in, and he binds to the sites on all these troponin.
Okay.
And when that happens, there's like a structural change that happens to troponin. What else is troponin bound to? It's calcium now, but also he's bound to 2 other things.
the actin and the tropomyosin.
Mm-hmm. When calcium comes in, it causes a change.
Okay.
The troponin kind of pull everything towards themselves. So since they're attached to, I'll try another one. They're attached to the tropomyosin, these sheaths that are covering the actin, and they kind of contract. They kind of pull everything in towards them, and they expose the actin, which allows
The myosin
the heads will go up and attach.
Because they will always attach if it's available.
Yeah, because their heads are attracted to the actin.
Okay.
So yeah, it goes up, and then it's gonna pull like this, right? It goes like this, and it shortens, and it pulls it. This is all on the other side too. I didn't
Yeah.
drop, but
Yeah.
and it pulls it together.
Okay.
And then when it's done, calcium leaves, relaxes
It
again.
relaxes, which also takes energy.
Yeah, it takes ATP to cause these heads to relax. Now, does that make sense? Is there any questions about that?
Yeah, I'm just trying to think of how like any of that's even going to be worded on the exam. Just like.
Probably the process. Why they could ask you, where does this calcium bind to actin? Does it bind to myosin? Does it bind to troponin?
Okay.
Things like that. They could do a true or false. Actin is attached to titin. Anything to test you on just being able to know this kind of
Okay.
diagram.
So the calcium binds to the troponin.
The subunit, yeah, one of the subunits
Okay.
on the troponin.
Got it.
Got it.
Mm-hmm.
Also, should we do the crossbridge cycle, or do you want to wait on that?
Yeah, we can. I mean, that's what we're going over in class now anyways.
So,
Oh, wait, sorry, the other thing. So,
Yeah.
like, T-tubules, what are those?
Okay, so T-tubules. So this is kind of zoomed in and looking on the inside of the muscle.
Yeah.
If we're looking at a muscle cell that's on the outside, like here's the plasma membrane, T-tubules are little canals. Say there's a hole right here, and you can see all the way through it.
Okay.
That's what a T-tubule is. It's
Okay.
like these holes that go through the muscles. So let's slice it sideways. Say this hole's going down like this, we can see through it. What happens? The T-tubule, here's a nerve,
Okay.
releases a signal. The action potential travels down here and then goes down the T-tubule.
And that's how it enters the muscle.
Yeah, well, kind of, yeah. Because there's a gate right here that it interacts with, which causes calcium to be released
Okay.
inside. We'll get to that.
And those are voltage gated.
Voltage gated.
Okay.
Well, yeah, kind of. There's two different gates right here. One's mechanical and one's voltage, but
Okay.
We can talk about that, too.
And then terminal cisternae.
what was it?
Terminal cisternae.
Terminal cisternae.
Or
I have to
get
look
to
at
it.
it. Pretty sure right here is the endoplasmic reticulum, sarcoplasmic reticulum,
Uh-huh.
and it's connected to two different gates. I think the cisternae is right there. Let me look that up. Just because I don't want to lie to you. Might be the next page. No, keep going. Wrote it down. Terminal cisternae. One second, I got to answer this.
Yeah.
Hello? Till when? So you'll get in Idaho Falls when? Two something. 2:20 is when you'll land in Idaho Falls. Okay. Okay, well, I mean, if it's 2:20, that'd still probably be fine. But, yeah. Just let me know when you land or whatever. Or when you take off, or when you're about to board. Alright. Bye. Gotta pick up my brother at the airport, come back from Brazil.
That's fun.
Did it?
So the only thing I have in my notes, I think it might have been the only thing from the reading. Well, it says sarcoplasmic reticulum is enlarged near the T tubules and forms terminal cistern.
Yeah, so that's what I was drawing. Here's the T tubule. Here's the sarcoplasmic reticulum, and this is a terminal cistern. That's what I thought it was, based on that term.
I mean, I usually just write things pretty much word for word from the book, so I think that's all.
Word for word from the book, and that's got to be accurate.
Yeah.
Okay.
Okay, so yeah, I guess we can just go over the. Well, wait, sorry. So what's the difference between fibrous and globular actin? I think that's the only thing.
Fibrous actin and globular actin. We never differentiated. Let me see. Sorry.
That's fine. I remember in my histology lab, like the day before we were going to take like an exam or something, at the very end of the lab, my teacher said something about them, but it was never discussed on like the videos and stuff either.
One's obviously spherical and one's linear, but
Yeah.
let's see. So actin. Under physiological conditions, actins exist as a long helical polymer, as fibrous actin, of a globular protein monomer. Oh,
So it just depends on the type of protein.
structure of the G-actin monomer. Binding region. So I think what it's saying is, okay, so all of these guys together makes up fibrous actin,
Okay.
but the individual monomers are called globular actin.
Okay.
Does that make sense?
Yeah, got it.
Together they make a fiber. The individual ones are around globular. Okay.
Okay.
All right.
Alright, so now we can, I guess,
Cross
just go
bridge?
over cross bridge is fine.
So can you kind of see how these two fit together? Like where would the Z disc be in this diagram right here? Would it be out here? Would it be somewhere?
Somewhere in the muscle fiber, I don't know.
Okay.
Just somewhere
Yeah.
in there.
So like all this could be kind of condensed down.
Yeah.
You put all in between here, right? Okay. Good. Just want to make sure you're kind of oriented.